Welcome to Brain Ponderings. I'm the host, Mark Matson. Today, my guest is Professor Martin Picard. He's Associate Professor of Behavioral Medicine with appointments in psychiatry, neurology, and the Aging Center at Columbia University, just north of Central Park in New York City. And he directs the Mitochondrial Psychobiology Group. And kind of the main point of my conversation with Dr. Picard, Martin, and his work has to do with the fact that mitochondria don't just produce energy for cells. They can divide, they can be destroyed, degraded in a controlled manner, process called mitophagy. They They signal to the nucleus. They receive signals from other cells and intracellular messengers. And it's very interesting, the recent explosion of work on mitochondria and all the things they do. And we'll also talk with Martin about neurological disorders. He's very interested in psychiatric disorders and the role of chronic stress and the roles of mitochondria in maybe mediating some of the adverse effects of chronic stress on mental health. So welcome, Martin.
Thank you, Matt.
And are you from Canada originally?
I am from Montreal, just south of Montreal, the French
You've got
part.
Canadian accent. I'm from Minnesota.
Oh, lovely.
So I don't say oot too much, but I have kind of a Minnesota accent, I guess. And
Yeah, nice.
I played a little hockey when I was young, and
Oh, love it. On the pond, on the frozen lake.
yeah, absolutely, and outdoor rinks.
Way to go.
And did you play?
I did, yes, I did all through high school. Hockey was my thing, and then I I didn't develop the physique of a Canadian hockey player.
I wasn't good enough to make a high school team, but one of the kids in my high school, Eric Strobel, played on the 1980 team, the Miracle on Ice.
Oh, nice.
Team. So I have that kind of connection. But back then, like the college team in Minnesota and elsewhere in the East, they're mostly from those states. Now all the Canadians are invading the United States college hockey, and the best teams now have Canadian players. So anyway, let's get to talk about mitochondria, but it's very important in hockey or any physical exercise, but it's also important in the brain, controlling brain function, even things like learning and memory, synaptic plasticity. Okay, so you have a really broad background because you learned a lot about mitochondria, I guess, early on, but then you did a postdoc with Doug Wallace, who was pioneer in these mitochondrial DNA mutations that cause these terrible disorders. And then from the stress end of it, you trained with the late Bruce McEwen, who is a pioneer on understanding how bad stress affects the brain. And there are bad stresses and good stress, I would propose. So anyway, can you talk about some basics of mitochondria?
Yeah, sure. So mitochondria are little living creatures. I think that's kind of a really good starting point because unlike other parts of biology, like that are inert, you're like, the genome is like this inert thing. Unless it's moved by energy, the genome just sits there, right, with ACTGs, methylated, or like other modifications. But unless, it's moved by energy, it doesn't do very much. Mitochondria used to be bacteria, I think, as most of your listeners will know. They have their own DNA. They have this double membrane like other bacteria, and they have a whole life cycle, inside the cell, where new ones are born through something called mitochondrial biogenesis, the genesis of new mitochondria. And then they diversify, just like cells do, right? You start with a stem cell, and then you end up with a bunch of different types of cells, like neurons and glia and cardiomyocytes and liver, hepatocytes, and white blood cells, and so on. So there's the same kind of diversification of mitochondria. So they're small living creatures that are genetically related to one another. Every mitochondria in the whole body share the same genome, just like every cell in the body shares the same nuclear, autosomal genes. And they diversify and specialize to play a number of essential vital roles. I mean vital here in the truest sense of the word. And they allow cells to do a number of things that otherwise cells could not do, like making heme, right? Heme is this critical piece of many enzymes have, heme components. The cell cannot make heme without mitochondria. And actually, if you block mitochondrial heme synthesis, the cell dies. You can make mitochondria that don't have, you can make cells that have mitochondria without mitochondrial DNA by playing some genetic tricks or hitting cells with some chemicals that deplete mitochondrial DNA. But cells will cells will need mitochondria to make hemes. Even if they don't have DNA and they can't transform energy, the cells will be glycolytic, but they need the mitochondria there, the mitochondrial DNA-free mitochondria to make heme and for other things like making.
From an evolutionary perspective, does that have something to do with iron and that bacteria are thought to have evolved, or the earliest life forms in the ocean floor in these hydrothermal vents that were really rich in iron and in hydrogen sulfide gas?
I don't know. Nick Lane would be the right person to ask about this. So
yeah.
I don't know what the evolutionary connection is, but going back to your question, mitochondria is this diverse group of small organelles. And unfortunately, fortunately, we have a single word, mitochondria. Singular is mitochondrion, right? Which doesn't, I think, pay justice and is kind of a holdup for really exploring and uncovering the diversity of mitochondrial functions and behavior.
Yeah, it's kind of like this saying the brain has neurons without saying, there are these pyramidal neurons in the hippocampus that use glutamate as a neurotransmitter, and they, serve this function. It's the same for mitochondria within an individual neuron. Neurons are incredible. We did some work just imaging mitochondria in living neurons in culture and following them over the time, from the time the neuron's born, grows axon dendrites, forms synapses. And they start with just a few mitochondria. And just over a period of a few days, all of a sudden you've got scores, even hundreds of mitochondria, and they're moving, they're present. It makes sense. You've got to have energy available all over, but it's quite remarkable.
Yeah, you have to have, the ATP synthesizing units, the mitochondria, where you need energy. Also, you need very high capacity for buffering calcium and maybe making reactive oxygen species, which the axonal mitochondria probably do. And so even you raise like a really good example. You have this single little neural stem cell progenitor, and then it differentiates as I'm going to be a neuron, and then boom, it starts to differentiate. Then it grows a bunch of one axon and then a bunch of dendrites. And a single pool of mitochondria, we could think of as a stem, stem-like mitochondria, now need to differentiate into axonal mitochondria, dendritic mitochondria, which have different proteomes, and they have different, functional capacities, and then different morphologies. Even in the living mouse brain, we imaged mitochondria in different parts of neurons, and it's remarkable. The dendritic mitochondria are extremely long, multiple branches, and multiple microns long. Like some of these mitochondria are, their length is like longer than multiple cell bodies. And then in the axons, they tend to be small little stubs, and so they're very different in the same cell type.
And you mentioned the proteome, which is the complement of proteins in the mitochondria. So my understanding is mitochondria have at least 1,000 different proteins. Most of them are encoded by genes in the nucleus. Only some, and particularly 13. Those involved in the energy production, are encoded by the mitochondrial DNA.
Yeah, only 13 of the, I think most people would say 1,100, 1,200, maybe 1,300 different mitochondrial proteins, and only 13 of them are mitochondrial DNA encoded.
And the mitochondria have two membranes too, which is another interesting feature of them. It's kind of a, I don't know, balloon within a balloon. I don't know how you want it. And the inner membrane, like, it's kind of convoluted, so that its surface area is quite large, actually.
Yes, exactly. I just received
Oh,
this.
yeah, good, good.
Yeah, and so here is the inner mitochondrial membrane, right? That has this beautiful shape, and then the outer membrane is on the outside here. So yeah, they're gorgeous. And then all of the action, the reason we breathe, right? And you could say the reason over hundreds of millions of years we have developed lungs and cardiovascular system and blood vessels is to bring oxygen to your mitochondria, right? So it might have been the mitochondria actually that pushed for evolving these physiological systems that actually feed them. And respiration happens right here in those cristae. This is where the oxygen, we breathe to feed mitochondrial respiration, which happens in those that are called cristae.
And then the carbohydrates and fats and proteins we eat, they can be used to produce ATP, the energy. ATP, we refer to it like as energy currency, but it's essentially the energy in the phosphate bond. So it's adenosine triphosphate.
Mm-hmm.
And the energy in one of those bonds is used to drive all sorts of processes. It's in neurons to extrude sodium and calcium after they've been stimulated, but pretty much everything that goes on requires energy. Moving the mitochondria actually requires energy.
Yeah, all of those beautiful movies you see, animations, I should say, where you have like the walking, little actin,
Oh yeah.
dynamine, or the motor proteins walking on the microtubule, like every little step, right, burns an ATP molecule or GTP. And there are many different energy currencies. ATP is one, GTP is another one, NADH, NADPH. So there are multiple forms of chemical energy that cells have the ability to transform between each other, but then also to leverage directly to power enzymes. And what I think the key to why mitochondria were allow so much complexity, right? And in multicellular organisms that have mitochondria that evolved, multicellular, multi-organ, nervous system bearing bodies, might have to do with the fact that mitochondria can take those chemical substrates, right? Like you say, the carbohydrates, the proteins, the lipids, and then mitochondria break those up and basically have the ability to rip off electrons from those carbon, backbones, flow those electrons, and then at every step as the electron flows, harness some of that energy to build an electric charge.
Yeah.
So effectively, this is close to magic. You transform chemical energy, right? Like chemistry into electricity. Like you go from a material form of energy to an immaterial form of energy. And electricity is one of the most versatile forms of energy that neurons use electricity across their plasma membrane as to store energy very quickly dissipated, right, to conduct action potentials. So once you have electricity, like the mitochondria is charged, now you can use it to power the ATP synthase and make ATP. You can also use this to pump calcium in. You can also use this to make reactive oxygen species, or to make heme, or to make precursors for DNA synthesis, or to power Krebs cycle, doing other like biosynthesis. So I think that maybe part of the magic of mitochondria is their ability to take chemistry, turn it into a much more versatile form of energy, electricity, and then that's used to make ATP, but then so many other functions.
Can you talk a little bit about I guess I would say the life cycle of mitochondria, right? You mentioned they can divide, but since they're like a major focus of free radical production, so in the process of generating ATP, the superoxide oxygen radical is formed. It can be damaging, but there's antioxidant enzymes, superoxide dismutase 2, for example, that convert that to hydrogen peroxide. But anyway, it's a major source for free radicals. And mitochondria, just during their normal aging and so on, accumulate a lot of damage. Oxyradicals damage DNA, proteins, lipids.
Yeah.
So can you talk about kind of the turnover of mitochondria in the
Yeah. So
healthy state?
yes, every biological system accumulates damage over time because of the natural imperfection, the inescapable imperfection of life. Anytime there's something that jiggles, right? If there's energy in the system and there's Brownian motion, and especially if you have a living system with like a ton of energy flowing through it to keep it alive, there's bound to be imperfections. And then I think that most people understand this as damage accumulation as the driver of biological aging.
Yeah.
That seems to be inescapable in most breathing creatures. Vadim Gladyshev has talked about the deleterium and the accumulation of damage, and so there's a whole field there. So mitochondria experience very high energy flow, right, with all of the electrons flowing that we talked about. And then if you have free electrons flowing, there's a chance that some of those electrons are going to leak out of the system, react with oxygen, and then make reactive oxygen species. Reactive oxygen species production by mitochondria seems to be a key signal for cells to divide, for example. And it's, key signals for, to keep normal, healthy cellular life. So mitochondrial reactive oxygen species production or emission seems to be one of their very important functions. That can get out of hand, especially if there's a lot of food substrates coming in, a lot of carbon backbones that are being entering into the Krebs cycle, and then you're ripping off all the electrons. So then the systems become all reduced, right? Like the NAD molecules have gained an electron. There's a lot of NADH, very little NAD. So the system is highly reduced. This enforces electrons very forcefully through the electron transport chain. And then the whole chain gets reduced because on the other hand, if there's no energy demand, the ATP synthase doesn't turn. So then you end up with a very high charge, electrical charge in the mitochondria. And this very high electrical charge is like having, a battery charged to the max and you never use it. In most, battery chemistry, that's a bad thing to do. And the battery actually degrades and, gets damaged by this. So if you have an electric car, it's recommended you don't charge it up to 100%. You charge it to like 80 for daily driving, because when your battery is charged too much, it puts a very high load on the membranes and on all of the systems. And chemically, what happens in mitochondria when they're supercharged and there's a lot of food coming in, but there's not of energy demand, there's all the system is highly reduced, and then a bunch of electrons start to leak out and react with molecular oxygen, creating the superoxide and ions, and then that's where damage happens. So this condition, people have referred to as metabolic oversupply. And then this is like the equivalent of just eating too much, even when you're not hungry, and never going into undersupply, like calorie restriction.
Like fasting.
Fasting, exactly. So if you never fast and you just constantly overfeed your system, this trickles down ultimately to your mitochondria, and then the mitochondria become, overly reduced, and then they start to make more reactive oxygen species, and this contributes to accumulation of mitochondrial DNA mutations, deletions, and then mitochondria are forced to, be degraded more quickly. And if you can't do that through the life cycle of mitochondria, then probably the health, quote unquote, of mitochondria degrades. So the life cycle of mitochondria is there to keep the mitochondria, healthy by growing the young healthy mitochondria into bigger ones, and you separate them, and the older fragments or the older mitochondria eventually get degraded into little pieces, amino acids, nucleic acids, and then those products get reused to build the new mitochondria. So the old ones die out, the new ones give rise to more young progenies.
Yeah, the cell has a mechanism to recognize a mitochondria that's not functioning well, and then move it to the lysosomes.
Correct.
Mitophagy. But I saw this was a long time ago. I can't remember the paper, but my understanding is that it's very interesting. Mitochondria apparently can segregate damaged molecules, damaged DNA or proteins to like one end before they divide. And then so that when they divide, the whatever you say, one daughter mitochondria is damaged and goes, the other one is more pristine. It's really a cool mechanism within the mitochondria, apparently.
Yeah, this is quality control, in a single organelle. It's amazing, and I think the molecular mechanisms for this are still being worked out. And maybe the paper you're referring to was this science paper where they discovered that a stem cell will butt off, a daughter cell that will then differentiate. The poorer mitochondria, the less high quality mitochondria, will be in the daughter cell. The mother stem cell that remains, the stem keeps the healthier mitochondria, and that might contribute to, maintaining a healthy population of stem cells. So
Yeah,
there was
that's
asymmetric division of
asymmetric. Yeah, that's really amazing. One thing that we didn't mention is that every cell in our bodies and brain, the mitochondria originate from the mother's egg,
yes.
right? So it's kind of a strange genetics of, so in these mitochondrial, can you briefly talk about some of these mitochondria? There's like Leber's, or I can't remember all the mitochondrial mutation disorders,
Yes.
but they're very terrible.
Yes, so the rare genetic mitochondrial diseases or mitochondrial disorders. I think the one you were referring to is Leber's optic neuropathy,
Yeah.
LHON. There's also a disease called MELAS, MELAS, that stands for mitochondrial encephalopathy, lactic acidosis,
Mm.
and stroke-like episodes, which is a long name to say you have elevated lactate, your muscles don't work well, and your central nervous system, your brain is headaches, migraines, and so on. So multiple systems become affected, and Doug Wallace and Ian Holt published 2 different papers in 1988, describing the first mitochondrial DNA deletion, point mutation and deletion that were the cause of Leber's and the cause of myopathy, genetic mitochondrial disease. And since then, I think, at this point, have over 300 point mutations that are known in the mitochondrial DNA. Mitochondrial DNA is 16,569 nucleotides. So they're all, numbered, and people refer to the mutations by the number.
Yeah.
Because it's manageable kind of intellectually. But there's about more than almost 400 now, I think, point mutations that are known to cause or have been associated with complex, multi-system diseases, or very local, like eye disease, losing eyesight, like in Leber's optic neuropathy. And those are caused, by point mutations or deletions of mitochondrial DNA. In some cases, they're inherited. So the mom will have, every cell has hundreds, thousands of mitochondria, and then therefore hundreds, thousands of mitochondrial DNA copies, which are little circles. And let's say mom has 10% of her mitochondria have this mutation, and mom is fine, right? And then she has a baby, and then the baby now has 50% of the mutation. So just by segregation and stochasticity, there can be, from one generation to the next, a large expansion of the heteroplasmy, the mixture of mutant and normal mitochondrial DNA. And then at higher percentage, some mutations now start to cause symptoms. And you start as a single egg, like you said, but then this egg, multiplies and then eventually segregates into different tissues. So some people have high levels of mutation in the nervous system, right? Like in tissues derived from ectoderm, but very low levels in the muscle. And then some other people have very high levels in the skeletal muscles and the heart, but low levels in the brain. So there's this heterogeneity in the distribution of mutations between tissues, and then between people, of course, and then between cells in a given tissue. You have, like, if you take a muscle biopsy, you have some cells with 100% mutation, the cell next to it 0. So there's heterogeneity at multiple levels.
And you mentioned the brain, the heart, and muscle, and neurons. These are all excitable cells, right? And they have a high energy demand. So intuitively, you'd think they might be more severely affected than other cell types that don't have this high. energy.
Yeah, exactly. These are the tissues with the highest mitochondrial content. Like if you ask like how many mitochondria are there, what part of the body has the most mitochondria? It's in the brain, in the heart, and the muscle. There's a few other organs that have a lot as well, like the kidney and the adrenal glands are packed with mitochondria, whose business is not to make ATP, but to make steroid hormones. Mitochondria is,
Yeah, yeah.
I didn't know until a few years ago, mitochondria are the source of all steroid hormones. It's amazing. The most
My
power-
my master's and PhD were both. My master's degree, I was studying adrenal steroidogenesis,
oh, cool.
and then yeah, my PhD, I was studying a neuroendocrine system that controls molting in crabs, and the molting hormone in crabs come. But
Steroids.
we're right. The early steps. These are all steroid hormones. All come from cholesterol.
Uh-huh.
The first step, or in some cases, a couple steps, in their conversion to the steroid occurs in the mitochondria. It's
Yeah.
very intriguing. But uh, oh, what was I going to say? Um,
Well, just
right.
to
Oh,
just
I know
more.
what I was going to say.
Yeah.
High concentration of mitochondria. Brown fat.
Yes.
Talk about brown fat's really interesting because say mice, lab rats and mice, they don't shiver.
Mhm.
They have a lot of brown fat, and the brown fat produces heat in the mitochondria. There's this uncoupling of the oxidative of the electron transport chain, essentially. And
Mhm.
instead of producing ATP, they can produce heat. And cold, when the animal gets in the cold, then it turns on the heat production. It's very interesting.
Yeah. Yeah. If you take a temperature probe, right, and you light up the cell, you ask like where in the cell is the heat coming from? I mean,
Uh,
that's a pretty valid question. And in the body, you can say, where is the heat coming from? Why do we have body temperature? You know, it's 37 Celsius, 90 some Fahrenheit. The answer is the heat comes from mitochondria. And the reason we are warm-blooded is because our mitochondria are hot. And there's a series of papers. I think there are 3 good papers on this in the last 2, 3 years, 4 or 5 years now, showing that mitochondria actually operate around 50 degrees Celsius, which is what's 50 Celsius in
wow.
Fahrenheit? It's very hot. It's
We
way
would die
above.
if we're out in that very long.
Oh yes, yeah, we would definitely die. But somehow it seems mitochondria operate, at much higher temperatures than the rest of the cell. So in a way, I think they're the little furnace um, of
Well,
the cell.
that's pretty interesting, isn't it?
And this is particularly true in the brown fat.
Actually, I hadn't seen that. So they have ways to measure temperature with that spatial precision down to the submicron level.
Yep, with spatial resolution, they're imaging based probes, so you can bring them down to single organelles, and it's pretty clear that mitochondria are pretty hot. And there's good biochemistry also. If you isolate mitochondria and you look at the isolated enzyme activity for complex 4, for example, which is the respiring component in the electron transport chain, and then you do a titration of temperature. You say, okay, what's the activity of complex 4 at 10 degrees Celsius, and then at 20, and then at 30, and then 37, which is body temperature, and then at 40, and then at 50 Celsius, and then 60 Celsius? You would think that the ideal temperature will be 37 because this is kind of the optimal physiological temperature. Turns out it's more like 50, 55 Celsius.
Huh.
And the enzyme doesn't, denature and degrade until much warmer temperatures. So that's, one of the pieces of evidence to suggest that really mitochondria very likely work at very high temperatures. And then they are probably the source of heat for the whole body. And brown fat have learned to leverage this to, regulate body temperature of the whole organism.
Okay, let's dig into your own work, which is really intriguing, exciting. You are essentially establishing a brain atlas of mitochondrial diversity, and you're applying your knowledge and methods. You came up with, I think you came up with this, I don't know, mitochondrial health index.
Mm-hmm.
And so can you talk about those two things, what you're finding in terms of diversity, and then how you're using the mitochondrial health index, because this becomes very relevant to human health and disease.
Yes, and maybe I'll talk about the health index first, because
Okay.
that led us to profiling the brain mitochondrial diversity. The mitochondrial health index (MHI) is a bit of a misnomer. So because mitochondrial health is very imprecise, and but the idea is let's quantify the energy transformation capacity, not on a per cell or per tissue basis, but on a per mitochondrion basis. So we're asking how specialized and how good are these mitochondria at taking substrates and transforming those into electricity. So that's pretty much what this mitochondrial health index does. And it's a very simple multivariate construct. So we measure the activity of the electron transport chain enzymes, complex one. There's complex 1, 2, 3, 4 that transport electrons and respire, and then complex 5 does the ATP synthesis. So we have good biochemical methods to measure complexes 1, 2, and 4 that we've been able to scale in 96 well plates, to get a little technical. So we can do hundreds of samples as opposed to like dozens of samples. So that increased our throughput dramatically by 2 or 3-fold. And then by measuring complexes 1, 2, and 4, we're measuring different parts of the energy transformation system. Then you can take those, mean center them, and then add them up as a numerator, right? So then you're measuring more or less the same thing in 3 different ways, aggregating this. This is energy transformation capacity. Now what you can do is on the same sample, which can be white blood cells, can be a piece of brain, can be a muscle biopsy or whatever, cultured fibroblasts. Then on the same sample, you measure citrate synthase, also a Krebs cycle enzyme, which is a marker of how many mitochondria are there. What's the mitochondrial mass or mitochondrial content in this sample? So per cell, citrate synthase activity, and then mitochondrial DNA copies per cell as well. And citrate synthase and copy number are both imperfect, not great markers of mitochondrial content, but they're still, together, you average them, mean center, add them up together. This gives you, as a numerator,
Yes.
how many mitochondria are there. So then you have at the, denominator. At the numerator, you have energy transformation capacity, and then denominator, you have mitochondrial content. So here you have a situation where you could have 2 different samples, 2 different people, let's say. They both have 10 units of energy production capacity, right? So the numerator is 10 units. But then person A achieves these 10 units with 5 units of mitochondria, right? So then you divide 10 by 5, you get an MHI, mitochondrial health index, of 2.
Yeah.
So that's person 1. Person 2, now 10 units, also same, right, on surface. But then you find out that this person, to achieve 10 units, they need 10 units of mitochondria. So 5 times more mitochondria. 10 divided by 10 equals 1. So the MHI of person 1 is 2 versus MHI of person 2 is 1.
Yeah.
So now you can say the health or the energy transformation capacity of person 1, even though they both have 10 units of capacity, this is achieved through healthier or more capable mitochondria. So that's the idea behind the MHI. And recently, my colleague Eugene Masharov, who's a chemist by training and an engineer at heart, and he's a neuroscientist by trade now. he helped us think about this a bit more biologically, and we realized that if you scale the mitochondrial mass, the cristae, like the enzyme activity of the electron transport chain scales with the surface area of the inner mitochondrial membrane. The mitochondrial citrate synthase, which is in the matrix, free floating in the matrix, and the copy number actually scales with the volume of the mitochondria. So we've made a few little mathematical adjustments to the MHI, and now we call this the MRC, the mitochondrial respiratory capacity.
Yeah, so you can have a situation where if you have a lower or worse, if you will, mitochondrial health index, the cells in that tissue sample may have a lot of not well functioning mitochondria, maybe even accumulating. And in Alzheimer's disease, there's really good evidence from Randy Nixon, right? He's up there in New York, that you get the accumulation of dysfunctional mitochondria. But they're not producing ATP very well, but they're generating a lot of oxygen-free radicals.
Yeah. So this could be, a de-differentiation or like a recalibration of the mitochondria. So now they've become really good at reactive oxygen species production and signaling. Maybe the neurons are like needing more of this trophic factor, or there's something broken. And at this point, I don't think we've been thinking about the system as a dynamic recalibrating,
I see.
plastic
Yeah.
system. So I'm not convinced that for the term mitochondrial dysfunction, I think we should banish. And because mitochondria have multiple functions, right? So if we say mitochondrial dysfunction, it makes it sound like they have a single function and therefore a single way to become dysfunctional, which is not the case.
Yeah.
So
you had a nice article on, kind of a perspective on mitochondrial nomenclature and among scientists, and I agree with it. I'm in the description section. I'm going to put links to a number of your excellent review articles, and including that one. Now,
okay.
Why don't you talk briefly about the impact of environmental exposures or life experiences on mitochondria? Because we've done a lot of work on effects of exercise, intermittent fasting, intellectual challenges on neuroplasticity, and have some evidence that these kind of good challenges are stress, I guess, is a good way to, in Selye's terms, good stress and bad stress. We and others now have evidence that these good stressors can, kind of like in muscle cells when you exercise them, can stimulate mitochondrial biogenesis. And we had evidence that we had this one nice paper where we use RNA interference to knock down levels of this PGC1 alpha, which is a master regulator mitochondrial biogenesis. And then we showed that mitochondrial genesis is necessary for both the formation of neural circuits and developing neural circuits, and in the maintenance of circuits in the adult. We had a Nature Communications paper on that. But anyway, you've focused a lot, I guess, because of being with Bruce McEwen, on bad stress and something called allostatic load.
Yes.
Can you talk a little bit about what you're thinking? I'm retired, so I'm not going to be doing any experiments, but I'm going to try to pick your brain on see where you're going with this, how you're going to study mitochondrial diversity and kind of link it to normal brain plasticity on the one hand and neurological disorders on the other.
Yes. So a few things. Once we have an MHI/MRC, metric, now we can start to apply this to ask questions that are relevant to to human life and to psychobiology. We are psychobiological creatures. Our life is full of subjective experiences that are, not visible under the microscope, or you can't run in a gel, or you can't sequence them. So this is the psychological or the subjective, dimension of human life. And then there's like the biological reality of our cells and neurons and all of this. And we're interested to understand how these things relate to one another. And our research program is centered around examining potential links between the subjective human experience and the bioenergetic processes that happen inside our mitochondria and inside, our cells. The brain is a big part of this. Like, there's a lot of things that happen in the brain, integrating information and then producing thoughts and emotions, and that drives physiology. And we know the brain is the most energetic organ by mass, of the whole body. So there's something a little mysterious that's happening there in the brain that's consuming a ton of energy, even when you're not doing anything and just resting. So we've applied this MRC-MHI approach to define and produce the first systematic map of mitochondrial diversity and mitochondrial energy production capacity in the human brain. And for this, we took a postmortem, a section of brain from someone who died, quickly of a heart attack, a non-brain, related issue. And then very rapidly, we're brought into the hospital. The brain was sectioned, frozen. So now we're 10 years later, and then I speak with, Eugene, my colleague, and I think, would it be amazing if we like knew where the mitochondria are in the brain and like whether they're different or are they all the same in the cortex and the subcortical nuclei, different parts of the cortex that neuroscientists really care about mapping, and do they all have the same types of mitochondria? And at the same time, we were deploying the MHI for the first time at scale in the mouse brain. And Ayelet Rosenberg, a student in the lab, took on the challenge to manually punch mouse brain regions in dozens of mice, and then 17 different brain regions plus non-brain tissues. So I think Ayelet analyzed almost 600 little samples, which was our biggest MHI study so far. And then we learned in the mouse brain that there are very different types of mitochondria that specialize in, the orbital frontal cortex in the front of the brain versus the hippocampus, dorsal versus ventral hippocampus have different types of mitochondria, and the putamen and the cerebellum and the brainstem and so on. So in mice, we were really surprised to see this beautiful molecular and bioenergetic diversity of mitochondria. And then we want to know, is this the case in the human brain? So Eugene, my engineer thinking colleague, he wrote code to, the engraver, a CNC cutter. You can like engrave a heart, in a piece of wood for your mom's birthday, for example. Yeah, so these you can buy something on Amazon for $300, and then it's a drill bit basically that's controlled on 3 different axes.
Oh.
So you can have it engrave a nice mitochondrion in a piece of plastic or wood. So Eugene basically programmed the CNC cutter to operate in the cold at -20 in a freezer room, and then to guide the little drill bit to engrave a grid, little bunch of little squares at fMRI resolution. So functional magnetic resonance imaging that people do if you want to image a human brain.
Nice.
Then what the scanner, the big MRI instrument does is it sections the brain into little cubes, and slices this way, and then you end up with, let's say, 3 millimeters by 3 millimeters by 3 millimeters. These are called voxels.
Yeah.
In the neuroimaging world. So Eugene produced this, the algorithm and software hardware approach to turn a frozen piece of human brain into physical voxels of the same resolution as fMRI. So then he was able to turn a hemisphere of a healthy human brain into 702 little voxels. And then we just deployed the MHI MRC platform on this, and then voila, we have systematically mapped the mitochondrial energy production capacity and diversity.
Wow. And now there's so many things you can do. You can imagine there are autopsy programs where they have brains from, say, a patient with Alzheimer's disease, a patient schizophrenia or whatever, and age match controls, and so then you could actually compare, and they've had fMRI done on them.
Yes.
You can compare that fMRI in the living person to the mitochondrial. What about proteomics then? You can do that too?
Yeah, you can do this on each voxel, right? But then each voxel, we found out, we took a few, of those voxels across the 702 voxels. We took one from the cortex, one from the hippocampus, one from the putamen, and one from the corpus callosum. And then we did single nucleus RNA sequencing to try to profile the mitochondria, but in a cell type specific manner. And this is where things get really interesting, because different cell types have vastly different mitochondrial phenotypes. So when you take a little chunk of brain, a little physical voxel, you mash all the cell types together, and then you get the averaged, right, of all the cells. But glutamatergic neurons versus excitatory inhibitory neurons versus microglia versus oligodendrocyte precursor cells, they all have very different molecular mitochondrial phenotypes.
Yeah.
So now we can map the mitochondrial specialization at single cell resolution, and this is what we're in the works of doing. In the next year or so, we'll be profiling mitochondria from hundreds of human brains where we have single nucleus or single cell RNA sequencing, and the resolution is amazing. The differences are astonishing. And we're discovering that the mitochondria in specific cell types appear to be responsible, for example, for producing vitamins for other cell types. So mitochondria are the main precursor of some vitamin pathways. And some cell types, their mitochondria, they don't have this machinery upregulated. Other cell types do. So
okay. So mitochondria are playing a role in intercellular signaling by essentially making signals. Well, steroids, we know that's
exactly.
true.
Steroids is a great example.
And why not be a lot broader than that? There's even some evidence that mitochondria can be moved from one cell to another. A colleague of mine at Hopkins, Dwight Burgles, had this nice paper where he showed that mitochondria can go from he's looking, I think, at the optic nerve. Mitochondria can go from the axon, inside the axon, to the glial cell, the oligodendrocyte that's wrapping around that. And, I don't know. Is that the other thing, I know you got to go pretty soon, but there's this evidence that bits of mitochondria can kind of bleb off and be released.
Yes.
And there's evidence that bits of mitochondria can be released from cells in these vesicles, small vesicles called exosomes or extracellular vesicles.
Yeah.
That could be it could be a signaling function. It could be just like a waste disposal thing too.
Yep. I think both options are viable hypothesis. And we've done a lot of work on cell-free mitochondrial DNA, which turns out to be cell-free mitochondria. And now we know in normal human blood and saliva, there is a lot of cell-free, free-floating mitochondria. Sometimes they're encapsulated in something, sometimes they're not.
Intact mitochondria.
Intact mitochondria, yeah. And there's a paper that says they're respiratory competent. So you take blood, you get rid of all the cells, just plasma, and there's like free-floating mitochondria there. So anyway, we could talk about this for a very long time.
Yeah. Right. But anyway, so there's evidence that bad stresses, chronic psychological stress, maybe through glucocorticoids or other mechanisms, can affect mitochondria. Is there evidence for that?
Yeah, we've known for decades that chronic life stress and early life adversity gets under the skin somehow and predisposes people to, illness and to short, lifespan. And we don't know how this happens. How does cell get under stress? How does stress get under your skin and all the way inside the cell, inside the epigenome, and, to change cellular aging? We don't know how this happens. The working hypothesis we're pursuing now is that mitochondria is actually a key player in this. Mitochondria are sensitive to stress signals. You get mitochondrial recalibrations, and then they start to produce signals that change the epigenome and that change, cellular behavior. So we see them as this dynamic, sensitive interface. And Carolyn Trump, an assistant professor in our group, has this paper under review now, for which there's a preprint showing that psychosocial factors. She uses this beautiful cohort where they ask people who were older individuals, how do you feel? And then people complete questionnaires on sense of purpose in life and social support and, positive psychological states and then negative psychological states. And then she asked, is this related? The way people are subjectively experiencing life, is this related to mitochondrial biology in their brain? So then these individuals who completed those questionnaires eventually die. Their brain goes into the biobank. You take a piece of the dorsolateral prefrontal cortex, do proteomics on it, do single nucleus RNA sequencing. So then you can profile the mitochondria at the proteome level and at the transcriptome level in a cell type specific manner. And Carolyn finds that there's a remarkable correlation between what people were experiencing, positive and negative psychosocial factors, and the biology of the mitochondria in the dorsolateral prefrontal cortex of the human brain. And this effect seems to be very cell type specific, and mostly the glial cells, mitochondria and the glia, seem to be most strongly, affected, or maybe they drive the subjective experience. We don't know at this point which direction this goes, but there's clearly psychobiological connection there with mitochondria in the human brain, and we've published some work in white blood cells. So this is an early area of research, but I think this has been a fruitful exercise so far.
Well, thanks for taking the time, Martin. I appreciate it. And there's a lot, if people want to read a lot more, as I said, I'll point them in the right direction. They can easily find you. You're not the famous Canadian chef.
No, I'm not.
You're the famous Canadian American neuroscientist. But anyway, yeah, it's an exciting time for you. I remember when I was, at where you are now, and you've got your lab going strong, and you've got a lot of good ideas, and I assume you've got some good funding. And
we
from
have
what I see, I expect it to continue. So
yeah, thank you. We have a great team, and it's a lot of fun, very stimulating. So thank you so much, and keep up the good work with the podcast. It's great.
okay, bye, Martin.
Bye bye.